Hi everyone, my name is Nicole Bermel and I'm filming this today on July 20th, 2025. I'm making this video to do my Module 4 application activity where I go over the movement of macromolecules within the cell. I chose 3 different macromolecules to go over today. We're going to be covering cortisol, glucagon, and actin. Okay, so these are the slides we're going to be discussing today, and like I said, we're covering the topic of movement of macromolecules. The first one we're going to look at today is cortisol, which is a steroid hormone. Cortisol synthesis begins with cholesterol, a lipid precursor molecule obtained from circulating lipoproteins, or it is synthesized within the cell. The production primarily takes place in the smooth endoplasmic reticulum, where enzymes initiate the steroidogenic process. Cortisol's biosynthesis is completed in the mitochondria, where specialized enzymes convert precursors into active cortisol through a series of oxidative reactions. After synthesis, cortisol is transferred to the Golgi apparatus in transport vesicles, where it may undergo further sorting or packaging. It is then enclosed in secretory vesicles and transported to the cell membrane, where it is then released via exocytosis into the bloodstream. As a steroid hormone, cortisol travels through the blood to act on distant target tissues. It plays a crucial role in regulating metabolism, the immune response, and stress. Unlike protein hormones, cortisol does not require surface receptors. It will just diffuse through membranes and bind to intracellular receptors to target cells. The next one we're going to be covering is glucagon. Glucagon is a protein hormone for export. Glucagon synthesis starts in the nucleus, where the gene for glucagon is transcribed into mRNA. The mRNA is transported to the cytoplasm via pores in the nucleus, where it is then attached to ribosomes bound to the rough ER. The mRNA is then translated and enters the rough ER. It will undergo folding and initial post-translational modifications. It then moves to the Golgi apparatus in transport vesicles, where it is further modified and sorted for export. Glucagon is packaged into secretory vesicles, which move towards the cell membrane. In response to stimuli like low blood glucose, these vesicles fuse with the membrane and release glucagon by exocytosis. Glucagon functions by binding to receptors on liver cells, stimulating the breakdown of glycogen into glucose. It exemplifies how protein hormones rely on surface receptors to export their effects on target tissues. The next one we're going to be looking at is actin, and actin is an intracellular structural protein. So unlike exported proteins or secreted hormones, actin is synthesized for use within the cell. Its gene is transcribed in the nucleus, and the resulting mRNA is translated by free ribosomes in the cytoplasm. Actin undergoes folding and assembles into filaments that become integral components of the cytoskeleton. It functions within the cytoplasm, where it helps maintain the cell shape, supports intracellular transport, and contributes to the cell's motility. Actin's role is vital in processes such as endocytosis, cytokinesis, and forming cell junctions. Its intracellular production pathway is simpler but no less essential than that of the secreted macromolecules. Finally, we have my work cited with the sources I have used to create my flowsheets. Thank you for watching.